Hey guys, this is cell growth and reproduction. So like everyone knows, we have two different types of cells. We have unicellular cells and multicellular cells. Unicellular cells are, as you know, they're cells that it's one cell, organisms that have one cell, and these organisms don't really have different types of cells. It's just like unicellular cells. But in multicellular organisms, we have two different types of cells. We have gamete cells and we have somatic cells. Somatic cells, also known as body cells, they carry out functions like in the body and stuff, and they undergo a process called mitosis. Whereas gamete cells are also known as sex cells. Somatic cells are also, they're diploids, and I'll talk about what a diploid is. Gamete cells are also known as sex cells. They are haploids, and there's two different types. In men, the sex cells are called sperm or pollen, and in women, it's egg. And these undergo a process called meiosis, which I'll make another video about. So let's talk a little bit about chromosomes. So chromosomes are structures that DNA is organized in. And it depends on what type of cell it is, it'll be different. So in prokaryotic cells, the chromosomes are arranged in a circular form. They're found in the nucleoid region of the cell, and prokaryotic cells only have a single chromosome. In eukaryotic cells, they are linear, that kind of look like this. They are found in the nucleus of the cell, and they're always in pairs. They're always in pairs. And one is usually from the male and one is from the female. And depending on how many pairs a eukaryotic cell has, that's what defines that species. For example, us humans, what defines us as humans is having 23 pairs or 46 chromosomes in total. But us humans, we have 23 pairs of chromosomes. How do chromosomes pair? Well, they pair by the types of genes found. What is a gene? A gene is DNA that codes for a specific trait by carrying the code for a protein. For example, we have one chromosome and we have from a male and one chromosome from a female. The way these are going to pair up is because this gene codes for hair, and it's going to pair up with the other gene that also codes for hair. And this gene that codes for the eye color is also going to pair up with the gene that codes for the other eye color. And because us humans, we have 46 chromosomes in total, some humans may have 45 or 47 chromosomes because something went wrong in the process. But if you have 47 chromosomes, you have Down syndrome. And what homologous chromosomes are a pair of chromosomes when chromosomes are paired. And remember when I talked about haploids and diploids? Well, haploids are cells that only have one chromosome out of a pair. Whereas on the other hand, diploids, the cell has a full pair of chromosomes. So somatic cells, they are diploids, and gamete cells are haploids. And fertilization occurs when the gamete cell, the sperm, fertilizes an egg, and it creates a zygote, which is a singular cell, and then that zygote will plant itself in the lining of a uterus and grow into a fetus. So now we'll talk about the life cycle of a cell. So usually the life cycle consists of the G1 phase or GAP1 phase, synthesis stage, GAP2 stage or G2 stage, mitosis and meiosis, and cytokinesis. G1S and G2 phase all create the interphase, and mitosis/meiosis and cytokinesis make the M phase. Some cells, if they need to, they'll stop growing and they'll become dormant, and that means they'll exit the life cycle and they'll enter something called G0 stage or GO stage. And The process of cellular division may happen faster or slower depending on what type of cell it is. For example, body cells like your skin or your hair grow a lot faster than brain and especially nerve cells. And like you can see this, or like your nail cells, like your nails are like your nails grow fast, your hair grows fast, but like other body cells like your bone cells, they'll grow a lot slower, they'll divide a lot slower, and that's why you grow taller over a longer period of time. And especially nerve cells, like after a while nerve cells stop dividing, which means that if you have any permanent damage to your nerve cells, then you're like hooked, and you have to suffer the consequences. And that's why it's really hard. Like you can't really do, like, if someone becomes like their spine gets paralyzed and their nerves are damaged, they may be paralyzed forever. So let's talk about mitosis. So as we spoke, mitosis occurs in somatic cells in multicellular organisms. And the first stage of mitosis is the G1 phase. And we have a cell right here. This cell, it has a nucleus. It has the full cell, a nucleus, and it has 2 pairs of chromosomes. So it has one chromosome is from a male and one chromosome is from a female. And it also has a centriole. And this is a stomach cell. And in the G1 phase, this stomach cell, its focus is it's doing its job. And for this cell, it's doing digestion, and it's growing. In the G1 phase, it's growing. And we can actually tell that this isn't a human species, like this cell is not from human species, because it doesn't have 46 chromosomes in total or 23 pairs. And so after a while, once the cell is done growing, then it's going to enter the S phase, and then it's going to divide into 2 individual cells. The reason why our cells divide is because if it gets, our cells take, they don't wait to get too big to divide, because when the cell is smaller, it's easier to divide. And yeah, so then after the G1 phase, it enters the S phase. So in the S phase, we see DNA replication. So each chromosome that we saw in the G1 phase was duplicated. And then these are going to get distributed into 2 individual cells later on in mitosis. After the S phase, the cell enters the G2 phase. And in the G2 phase, we saw how our centriole was duplicated. And not only that, but all the enzymes that need to help in cellular division, all the proteins, those are synthesized in the organelles, and all of those organelles are duplicated. I just didn't draw it, but there's duplication of every single organelle in the cell. And in the G2 phase, it's kind of like it's getting ready to undergo the division process. So how does the mitosis division process happen? Well, you can memorize it by an acronym, PMAT, PMAT. And the first stage is the prophase stage. And the first thing that happens in the prophase stage is that those chromosomes or chromatin, which is like the straight ones, the uncondensed ones, they condense, okay? And they condense by like curling up really tightly around, and they curl around things called histones. What are histones? Well, histones are families of proteins, and they act as spools for DNA. I don't know if you've ever seen a spool, but it looks something like this. If you like sewing, then you'll know what a spool is. And it's like a spool, and around the spool is where the thread is coiled around. And the benefits of these histones, which are proteins that the DNA is coiled around, the benefits of these histones is 1, that it compacts them, that it prevents tangles, and 3, it controls how much DNA will be expressed. So the 3 benefits of histones are 1, that it compacts, 2, it prevents tangles, and 3, it controls how much DNA will be expressed. Because it's yeah. And so these chromatin, they're going to condense around histones, and this is the first time that we can actually see these chromatid under a microscope, because when they condense, they actually become thicker. So that's when we can actually see it under a microscope. And they also condense because it's easier to divide. And so after the chromatin condense, each each chromatid, they form an X. They form an X by like the help of proteins, and they form a region called the centromere, which is this gray dot in the middle. And so they form an X shape. And now these are called sister chromatids. And then we also see how the centrioles start to move towards the pole. They start to get attracted towards the poles. And this helps in the other stages. And also in prophase, we see how the nuclear envelope dissipates. It starts to dissipate. And then and then that's prophase. So then it enters metaphase stage, where in the metaphase stage, we saw the centrioles radiate towards the poles. Well, now these centrioles, they're going to radiate things called spindle fibers. And spindle fibers, they're fibers that attach. So some spindle fibers attach to the sister chromatids. And these sister chromatids, we see they line at the equator of the cell. And then the spindle fibers attach to the sister chromatids at the regions of the centromeres. And some spindle fibers, they attach from centriole to centriole. They're the continuous spindle fibers. Like you can see how some of them, they just go straight from centriole to centriole. And this helps in anaphase. So in anaphase, which is the next stage, we see these spindle fibers, the spindle fibers that were attached at the centromeres, these spindle fibers start to get shorter. And when it starts to get shorter, it actually pulls the sister chromatids apart, creating, like on the bottom, these like V shapes and then these A shapes. And we see that these continuous spindle fibers, the one that went from centriole to centriole, those actually get longer. And they get longer because they add amino acids and proteins to them. Anyways, so the last step of like the mitosis part is the step called telophase. And in telophase, we see that these continuous spindle fibers, they caused the cells to elongate. And that's where it kind of initiates where this pinching mechanism is going to happen. And this pinching mechanism is actually a mechanism called cleavage furrow. It's referred to as cleavage furrow. And we also see how our chromatid decondensed, they uncondensed and created chromatin. So the cell kind of in anaphase, it kind of like split in half, and then these uncondensed. And then we also saw the reappearing of the nuclear envelope. But at the end of telophase, you see one continuous cell. You still see a continuous cell, it's just in an oval shape and it's being pinched. When it actually becomes two separate cells, it is in cytokinesis, where it's fully separated. The telophase, like the one cell is fully separated because the cytoplasm was divided and all the organelles were equally distributed. And these cells are called daughter cells. And then these daughter cells are going to undergo the G1 phase again, where they're going to grow and then become even more cells. So yeah, that is mitosis.